The last speaker in this session is Pierce Ogden.
Thank you very much, Eric. I'm excited to tell you all about what we're working on at Manifold, which was inspired a lot by gene delivery and AAV and trying to bring the methodologies of capsid engineering to protein therapeutics more broadly. And so our vision is to unlock the ability to deliver any medicine modality to any tissue of interest. So this could be antibodies, oligonucleotides, and enzymes, where we want to get them to specific locations, but we lack the ability to localize those medicines to where they can function. We believe antibody-like shuttles, which are small binding domains that bind specific receptors, will enable you to localize to specific tissues, but the problem is we don't have many of these agents yet. And our ability with in vivo focused design and measurement allows us to engineer and generate these shuttles. So I'll tell you a little bit about how we do that. But before, I want to describe why this is a challenging problem. Tissues are composed of many cell types. And if we zoom in and think about a specific cell, there are many receptors on the surface of that cell that represent opportunities to target that cell specifically. The majority of these have been not explored to date. And we really don't have the information to know which of these receptors, and more specifically, which epitopes on those receptors will be good targeting domains. And ultimately, this is something we need to test in vivo to really understand, because this cell is embedded in a tissue, which is embedded in an organism, which expresses 20,000 plus proteins. And ultimately, getting that modality to that specific tissue means you have to survive that complexity and end up in the right location. And so our platform allows us to do this, starting with computational target prioritization, looking at single-cell RNA sequencing and proteomic data to understand which receptors are expressed where. We then have the ability to use AI-guided protein design to design millions of these putative shuttles in a structurally focused way to hundreds of receptors, and then we are empowered via a very unique approach in vivo multiplexing to quantify thousands of these molecules across an organism. And so I'll show you some data of how we do this. Quickly walking through the AI-guided protein design, we've built an internal tool we call mBER that allows us to perform de novo binder design in a structure-aware manner. I won't talk too much about this. We've actually published this work, so you can read about it. And there's many others like Boltz, et cetera, that are performing specific or similar methods. But the power of what we do is we can do this at scale. We've designed millions of binders to hundreds of targets here, showing many targets in green and blue, and the binders in red that are binding those receptors. And importantly, most of these receptors are novel, so we don't know what they do. And then our power comes from being able to put all of these molecules into an animal in a living system and then assess where they go and then validate and qualify which of those receptors work. And so I'll walk you through a little bit about how we do this. We have an in vivo multiplexing approach. And so really what this allows us to do is take many of these agents, inject them into an animal, and then take out the various tissues and quantify which of those agents are present in a specific tissue, ultimately with the goal of making tissue-specific shuttles. We have two approaches to this. I won't get into the details, but ask me about it later. One where we can do a very, very high plexity, hundreds of thousands of molecules hitting hundreds of targets, and look at relative tissue enrichment. And then another method that allows us to really get fine-grained, highly accurate quantitation of drug-like molecules at the scale of 50 to 1,000. And so here's actually some of the data of taking those designs I was showing on a previous slide and putting them into a living system. I'll quickly walk you through this data. This is 157,000 shuttles we tested. What are displayed here on rows are a subset of those shuttles. The columns are the various tissues we're looking at, and the heat is the enrichment of that molecule into a specific tissue. And so you can see I've sub-selected a group of molecules here that have some tissue specificity. And you can see in one experiment, we've actually been able to look across the entire organism. This is a mouse, but and understand which molecules go to specific tissues. We can then take this data and follow up on it and validate some of these molecules. So here, taking a subset of these molecules from the brain and looking at, do they actually get into the brain? And so here in blue is one of these hits compared to a TFR shuttle, which is kind of the gold standard for brain delivery right now. And you can see that this molecule is substantially improved in its ability to get into the brain. This is kind of the AUC for this PK curve here, and so you can see it's much better than this TFR shuttle. And importantly, when this molecule gets into the brain, it can then engage a specific target of interest. Here we're looking at amyloid beta in red. Our shuttle is in green. This is compared to an unshuttled molecule where you see very little co-localization of plaque and antibody. Here we see a lot of co-localization, and this is just a confocal zoom in of that as well. And so we're expanding this approach to all tissues beyond the brain as well. We want to generate agents that can target specifically to any tissue and really any cell type of interest to cure or enable curing many diseases. And so I'll show you a little bit of this data looking at some other tissues. Here's a couple of lung shuttles that we looked at where we followed up and see that indeed these targets, when engaged, enable a molecule to get into the lung as compared to a negative control. And so we're following up on this now, looking at oligo delivery to lung for COPD and other diseases. And some other examples in other tissues, we're very interested in the adipose as well for cardiometabolic and obesity-related indications. There's a big opportunity here. And so similarly, we took, I didn't move the box. We took we took some of these molecules that bound the adipose and tested them actually in NHP to see if they enable delivery to NHP. And here we do and see that this novel target indeed enables a molecule to get into the adipose at a 50-fold increase as compared to an unshuttled version of the molecule. We've then taken these molecules and put oligos on them to see if we can knock down targets in the adipose tissue, and indeed we can do that very specifically. And so here, looking at kind of a dose response for an oligo conjugated to one of these shuttles, we see very specific and dose-dependent knockdown in the epiwatt, and importantly, we avoid other tissues like the kidney and the muscle that you might want to avoid for specific therapeutic indications here. And so, I hope I've shown you a little bit of how we're generating tissue-targeted medicines here and really exploring novel receptor biology using AI-guided design of proteins that hit those novel receptors with the goal of enabling any medicine to get to any tissue of interest. So thank you very much, and happy to chat.